Hello students, in this video we will learn about the human ABO blood grouping system, RH factor and the basics of blood transfusion or blood donation. So first, human ABO blood grouping system. Carl Landsteiner in the year 1900 discovered that human beings have glycoproteins called antigens, or it is also known as agglutinogens, on the surface of the RBC. Suppose this is the RBC, and on the surface of the RBC antigens are present or agglutinogens are present. And gamma globulin proteins in the blood plasma, that means this is the RBC which is present inside the blood vessels, which is flowing through the blood other components, and this is the blood vessel and this is the blood plasma, and in this blood plasma the protein, the globulin protein which is present, known as antibodies. So antigens are located on the RBC surface, on the wall of the RBC surface, and antibodies are also called as agglutinins, are located in the blood plasma. Now, two types of antigens, A and B, and two types of antibodies, A and B, are found in human blood. So this capital A and capital B for antigens, and this small a and small b for antibodies. So two types of antigens, capital A and capital B, and two types of antibodies, small a and small b, are found in human blood. Now, based on presence or absence of these antigens, human blood falls into four groups. Person with antigen A belong to blood group A. Person with antigen B belong to blood group B. Person with antigen A and antigen B belong to blood group AB, and person with no antigens belong to blood group O. This forms ABO system. Of blood groups. Persons with one antigen have natural antibodies in their plasma against other antigens. Persons with antigen A have antibodies for antigen B in their plasma. What I have mentioned, person with antigen A have antibodies, this is the structure of antibody, have antibodies for antigen B in their plasma, which is represented as, and this one is represented as anti-B. Person with antigen B have antibodies. Again, the same structure I'm drawing for antibodies for antigen A. That means they have a antibody A or anti-A in plasma. Person with both the antigens, person with both the antigens A and B do not have antibodies, and person with no antigens and person with no antigens no antigens no antigens person with no antigens have both the antibodies, that means anti-A and anti-B. Antigen A and antibody A are incompatible. Similarly, antigen B and antibody B are also incompatible. Antigen A and antibody A, I'm writing here anti-A, are incompatible. Similarly, antigen B and antibody B, here I have written anti-B, are also incompatible. It means when blood from different blood groups A and B mixed, when blood from different blood groups A and B is mixed, clumping occurs due to antigen antibody interaction. This is called antigen antibody interaction. This is called clumping reaction or agglutination reaction. I repeat, this kind of clumping reaction is known as antigen antibody interaction or agglutination reaction. In addition to A and B antigens, human RBCs also possess Rh antigen. Human population is divided into Rh positive and Rh negative persons. Above 85% of human population is Rh positive. Rh persons have Rh antigen, while Rh negative person do not have Rh antigen. Rh negative persons do not have natural antibodies against Rh antigen. What I mentioned, Rh negative persons do not have natural antibodies against Rh antigen. Rh blood group system, this blood group system Now I'm talking about Rh blood group system. So Rh system, here I'm writing Rh system or Rh blood group system was discovered by Landsteiner was discovered by Landsteiner and Wiener in the year 1940. In the year 1940. Rh antigen is formed by dominant R gene. So this is the structure of gene, and suppose this is capital R gene, and this gene is mainly responsible for the production of this Rh antigen. So the name of the gene is R gene, and this particular gene is responsible for the Rh antigen. Gene means the specific DNA segments, okay, and it is represented as capital R, and that particular segments which is known as capital R is responsible for the production of this Rh antigen. Therefore, a person may have genotype capital R or capital R small r. Okay, so a person, when they have the genotype capital R or capital R small r, that means he is Rh positive person, because capital R is the dominant form, small r is the recessive form. An Rh negative person, what about the Rh negative person? So Rh negative person's genotype is always smaller. That means recessive. So it is homozygous dominant form, that is reflecting Rh positive. That is heterozygous dominant form, one capital R and another smaller, indicating Rh positive antigen. And when both the recessive forms are present in homozygous situation, that means homozygous recessive, it is indicating Rh negative. That means that person is Rh negative person. Now when Rh negative blood is given to Rh negative person, when Rh negative blood is given to Rh negative person, the transfusion is successful. Similarly, transfusion of Rh positive blood to Rh positive patient has no problem. That means that time also transfusion or blood donation is successful. When Rh positive blood is given to Rh negative person, anti-Rh antibodies develop in the blood of recipient. That is the recipient, this is the donor. This is the donor, he is donating Rh positive blood to the Rh negative person. Okay, so anti-Rh that is anti-Rh antibodies develop in the blood of that Rh negative recipient, and this is known as immunogenic response. It is called immunogenic response. Immunogenic response. Immunogenic response. So what I've written, Rh positive donor is giving the blood to Rh negative recipient or to the patient, and due to that the immunogenic response develop inside the body of this Rh negative patient or recipient. This is the first transfusion. Suppose this is the first transfusion, and during the first transfusion, immunogenic response develop uh, due to the formation of anti-Rh antibodies inside the body of this Rh negative recipient. A second transfusion, this is the first transfusion. Now it is the second transfusion of Rh positive blood to such a person results in clumping of donors blood cells, because Rh antibodies already developed into recipient's blood react with Rh antigen. That person or recipient has anti-Rh antibodies. So in the second time, when Rh antibodies again introducing in this patient, then there is the clotting or clumping occur. For these reasons, while determining ABO blood groups, presence or absence of Rh factor is also tested. Rh negative persons should be given Rh negative blood only. So this is about the Rh system. Now move to the blood transfusion. Because of agglutination reaction, transfusion of blood between individuals of different ABO blood groups results in clumping. Transfusion fails, especially if large quantity of blood of different blood types is introduced. Either the antibodies of recipient blood destroy the RBCs of donor blood, or the antibodies of donor's blood hemolyze the RBCs of patient or recipient. In case of small quantities of blood are introduced, blood transfusion is successful only if the plasma of recipient's blood does not contain antibodies against donor's antigens, or donor's blood has no antigens. It means blood from O, here you can see blood from O can be given to persons of all four blood groups. Here it is written can donate to, that means O blood group can donate to A blood group, B blood group, AB blood group and O blood group. So it means the blood from O group can be given to persons of all four blood groups because it has no antigen. See, it has no antigen to react with recipient's antibodies, and therefore person with O blood group is called universal donor. Persons with blood group AB have no antibody. See, no antibody in their blood, so they can receive, here it is written, can receive from AB, B, AB or O blood group donor. So they can receive blood from any blood groups. Therefore, persons of AB blood group are called universal recipients. The person who gives blood is called donor. The person who receives blood is called recipient. For blood transfusion, the blood group of donor and recipient need to be the same or should match. They should be compatible. In incompatible blood transfusion, the antigen of the donor's blood, suppose this is the antigen of the donor's blood, antigens of the donor's blood are attacked by the antibodies of the recipient blood. These cause clumping of donor's RBCs inside the recipient or inside the patient's body. The antibodies in the plasma of a person do not react with antigen on the RBCs of the same person. It means those blood transfusion are the most successful where the donors and recipients blood groups are the same. So here the summary is given: blood group A and A blood group person on the RBC surface, he has antigen A and antibody B is present and can donate to A and AB blood group person and can receive from A and from O. B blood group person having the antigen B, antibodies anti-B, anti-A, and can donate to B and AB and can receive from B and O. And about AB and O already it was discussed. And this is about the location. The lower one is mainly reflecting about the location of the antibodies and the antigens. Okay, so this is the red blood cell and you can see the structures. These are indicating the antigen. Okay, and antibodies are mainly present in the blood plasma. So this is about the blood group, human ABO blood grouping system, RH factor and blood transfusion process. Thank you for watching this video.